everyone, and welcome to today's episode of our Discover Molecular Interactions webinar series. My name is Vivian, and I'm responsible for scientific communication at Bruker Biocenters, and I will be your host in today's webinar. Our topic today is the very fascinating connection between immunology and metabolism. For example, how can metabolites influence immune responses or also immune cell behavior? And our guest speaker today is Dr. Tomas Paulenda. Hi, Thomas.
Hi, everyone.
And he received his PhD from the Institute of Molecular Genetics at the the Czech Academy of Sciences in Prague. And he is now working on elucidating the therapeutic potential of immunoregulatory metabolites, for example, in infectious diseases and cancer. If you have any questions during his talk, please feel free to use the Q&A feed, or if you don't see that, you can also use the normal chat function. Or you could also raise your hand afterwards in the Q&A session, and I can unmute you, and you can talk to Thomas directly. And now, without any further ado, I will hand over to you, Thomas, and I'm looking forward to your talk.
Thank you, Vivian. I'm very happy to be here with you and be able to present our work when we found out about itaconate. And so to just jump right in, one thing we basically need to establish is where itaconate is coming from. And the fact of itaconate, one very interesting fact, is that it's a very restricted metabolite. So it's not actually present in all the cells, as we usually know for many metabolites, but it is specifically produced in activated myeloid cells, like macrophages or dendritic cells. And what happens in macrophages upon stimulation actually is that they undergo a very strong metabolic rewiring, and so they start basically producing energy in a different way to compared to normal cell. And one of the features is that their TCA cycle breaks in two points. One point is the production of itaconate from aconitate, which wasn't actually discovered that long ago. And this is catabolized by enzyme IRG1, which is responsible for production of itaconate. And really amazingly, itaconate is produced in huge quantities in activated immune cells, which have consequences for its function and also for our study. And the second breaking point that happens in the TCA cycle in macrophages is that itaconate is an inhibitor of succinate dehydrogenase, which is an enzyme, another in TCA cycle, which catalyzes conversion of succinate to fumarate. And what happens when you inhibit SDH? So if you inhibit the enzyme that is utilizing succinate, succinate is actually accumulating in macrophages in a long LPS stimulation. But when we remove the enzyme IRG1 that catalyzes production of itaconate, these macrophages are no longer accumulating succinate. So SDH inhibition is basically driven solely by itaconate in activated macrophages. And we believe that this inhibition of SDH will be the thing that is driving the immunoregulatory functions of itaconate, because what we see here is that in the inflammasome activation, which is a way how immune cells can be activated to produce cytokines, in this case IL-1 beta, IRG1 knockout, in absence of itaconate, we see more IL-1 beta produced by macrophages. So this suggested to us that itaconate is a negative regulator of immune responses. So it's an anti-inflammatory metabolite. And another feature of itaconate that was discovered in our lab is that itaconate is a mild electrophile and can modify cysteines. And in the beginning of itaconate research, people believe that itaconate, since it's a bicarboxylic acid, will not be able to get inside the cell freely. And so derivatives were introduced. And these derivatives are basically just modified itaconate that allows it to penetrate through the membrane. But what also happens is that these derivatives are much stronger electrophiles meaning that they are much more prone and much faster to modify cysteines and interact with them than the natural itaconate is on its own. And this turned out to be very important when we looked into the interferon production by macrophages. And so my colleague Amanda Swain discovered that when you pretreat macrophages with high quantities of itaconate for extended period of time, this is actually not a high quantity, it's something we expect to be intracellular concentration of itaconate, then they produce large quantities of interferon upon stimulation with LPS. So this is basically showing for the first time that itaconate can also be pro-inflammatory, not only anti-inflammatory. And when we do the same thing with derivatives of itaconate, so these that are strong electrophiles, they are actually doing the exact opposite. So they will be inhibiting interferon. So this already tells us that we can't use derivatives as a proxy for itaconate to study its biology because they may have very different effects on cells. And I would like to kind of say here that we actually think now that we gave the electrophilicity of natural itaconates too much credit for its function. And so we're basically coming here with two phenotypes at the beginning of our study. One phenotype is the interferon, where we see that pretreatment with itaconate leads to strong interferon production upon LPS. And we have also the inflammasome inhibition. And so I will focus now on the part of the interferon for this story. We also get back to the inflammasome at the end, but this talk will be primarily focused on the interferon. And so previously I showed you interferon production upon LPS, which is a one stimulation possible. There's also other stimulants like poly-IC, which is essentially an RNA analog. So this is going to be more related to viruses. And we see that even upon poly-IC stimulation, interferon is strongly boosted. I told you already that itaconate is an SDH inhibitor. And so to see whether this is purely driven by inhibition of SDH, we used malonate, which is another molecule very similar to itaconate that's also a known SDH inhibitor. And we see that pretreatment with malonate, to the same extent like itaconate, doesn't actually boost interferon at all. And we use these as an acid, so we basically just control the acidity of the environment. So with hydrochloric acid, the acidity is very marginal. So there we see no effect, no boost. So this is not SDH driven. And when we see this with the exogenous itaconate, so what about the endogenous? So now here we are looking at the itaconate that is produced by macrophages upon activation. And in poly-IC, we can actually see that 24 hours where we expect there to be high quantities of itaconate already present. There's a very nice difference in wild type versus IRG1 knockout macrophages. So these are macrophages unable to produce itaconate. And we see that lack of endogenous itaconate actually leads to lower interferon, which is consistent with our exogenous itaconate data. And when we reintroduce the itaconate into the system, so we apply it into media, actually it can rescue the itaconate, the levels of interferon, and also it can rescue the levels of itaconate. So here measured the actual itaconate inside the cells, which is now rescued by the exogenous in the knockout. You see there is nothing produced upon stimulation, and it is rescued by exogenous itaconate. And the last step that we wanted really to see was, will it have the same effects in vivo? And indeed it does. So when we treated mice with poly-IC, and after 6 hours, we looked at the levels of cytokines in their serum, we see that in absence of itaconate, there is actually a lower interferon production. And this is also true for poly-IC and also different stimulation which is directly targeting a cGAS STING pathway. So this is a STING agonist, and we see that in here as well, the interferon levels are lower in absence of itaconate in IRG1 knockout. And the exciting thing about DMXAA is that in a prolonged treatment, it's actually lethal. And the lethality in mice is purely caused by interferon. So if the mouse has high interferon induced by DMXAA, they actually succumb to this treatment. And when we knock out IFNAR, which is one of the most important receptors for interferon in the mouse, they are completely protected and they don't succumb to the treatment. And so if we look into wild type versus IgG1 knockout, we can see that the lower interferon in the IgG1 knockout actually protects the mice, and it's functional in this setting, and these mice actually survive compared to controls that with a high interferon actually succumb to the disease. So now we have like a complete set of observations. So we know that exogenous itaconate boosts interferon, endogenous itaconate boosts interferon, and it does it not only in vitro but also in vivo in mice. And so the question is, what is the underlying mechanism, how itaconate does this? And so first, of course, we looked at the interferon-inducing pathways in cells. One of them is MDA5 and RIGI, which is an RNA sensing pathway that is converges on a protein MAVs. And we can see that the cells lacking MAVs actually are still boosting interferon. This suggests to us that it is not driven through this pathway. But when we looked into the DNA sensing pathway, which is cGAS and STING, we actually see that the boost of interferon is lost in cGAS knockout or STING knockout, telling us that interferon is boosted through cGAS-STING pathway. And this is also confirmed by direct stimulation of macrophages. The DMXAA leads to higher interferon production in itaconate-treated cells. So it all nice fits together. And cGAS and STING are known to be dependent on mitochondrial DNA. We know that itaconate is produced inside the mitochondria. So we looked if the mitochondrial DNA is important, and we depleted it from the cells. And then when we looked into interferon production in the cells that have depleted mitochondria, the boost is much weaker, diminished compared to control cells. So this is mitochondrial DNA dependent. We do see that the mitochondrial DNA is released into the cytosol upon itaconate treatment. But the fact is that is also happening with malonate. So if you remember, I showed you that malonate doesn't boost interferon, but it still leads to release of mitochondrial DNA due to mitochondrial damage. So the important question is now that there must be something other than just mitochondrial DNA that is driving the boost, and something that is actually sensitizing STING signaling. And to kind of cut it short, we found that the thing that is different between itaconate and malonate is ROS. And when we looked into the general ROS and we depleted it with N-acetylcysteine, we see that dose dependently the boost induced by itaconate is lost. We see the same thing with endogenous itaconate. So if we treat wild type macrophages producing itaconate upon poly-IC with NAC, we decrease the level of interferon production to the level of IgG1 knockout. And there's pretty much no effect in IgG1 knockout with NAC. So depletion of ROS actually eliminates the itaconate mediated boost. And I mentioned that itaconate is produced in mitochondria, and that's probably where we will see the highest concentrations of it. So we also looked into the mitochondrial ROS, and we see the same picture. So if we deplete mitochondrial ROS using mitotempo, we lose the boost induced by itaconate. So this is ROS-dependent phenotype. And to measure which type of ROS, because there are many of them, which type of ROS can be driving this, we measured mitochondrial hydrogen peroxide. And this is the only one where we found differences, that you see upregulation of hydrogen peroxide by itaconate in mitochondria, but not in the case of malonate. And we confirmed it also using recombinant hydrogen peroxide probe that is expressed in mitochondria, and it's fluorescent when it interacts with hydrogen peroxide. We see that the levels of peroxide are elevated in HeLa cells, and it's not only elevated in resting state, but what we saw is that the decomposition by hydrogen peroxide is slower in presence of itaconate. So itaconate seems to be elevating the hydrogen peroxide levels, but slowing down the hydrogen peroxide removal. And hydrogen peroxide removal in mitochondria is driven by a number of pathways. We don't need to kind of go into detail in here, but it's very tightly regulated. And to find out kind of which of these pathways can be affecting it, and we still believe this can be basically caused by the electrophilicity of itaconate, we performed the protein-wide search of itaconated proteins. And here I actually went back to the paper and I found that we used Bruker instruments for these. So it's not only the Bruker biosensors that we worked, but we basically pretreated macrophages with itaconate or stimulated with LPS, and then extracted the proteins, digested the proteins, and measured the peptides obtained and searched for the itaconated peptides. And we identified a number of proteins, but one very important hit that we got from this analysis was PRDX5. So we actually saw that one of the peroxiredoxins of the system that decomposes hydrogen peroxide in the mitochondria is modified by itaconate. And so this is the spectrum. We see that resolving cysteine, so one of the key cysteines in the enzyme, is itaconated, which could affect its function. So let's see what happens if we pretreat the peroxiredoxin with itaconate, and we see that it's actually very strongly inhibited by itaconate treatment, but not by malonate. This is our control here. And the inhibition is dose dependent. So if you decrease the concentration of itaconate, the inhibition goes away. And it's not only mouse peroxiredoxin 5, but it's also human peroxiredoxin 5 that is inhibited in presence of itaconate. And we also tested the activity method that contains the full redox system for peroxiredoxin 5, where we have thyrodoxin reductase, and then the NADPH is utilized. Here we basically measure the activity as a rate of NADPH disappearance from the system. And here again, you can see that the activity of PRDX5 is inhibited by itaconate, and it's intact in presence of malonate. So now we confirmed that it actually inhibits the peroxiredoxin 5, but the fact is that the itaconation of the peroxiredoxin 5 is actually very small. So fraction of the itaconated protein is only 1.5% of the entire protein in the cell. And so this is very low to explain our observed phenotypes and observe the inhibition. And so again, I think we gave electrophilicity too much credit in the beginning. And so we basically were looking for something else that could explain this. And here is where we found Bruker biosensors, and where they really helped us immensely, because it wasn't easy to find a method how to prove the protein small molecule interaction in case of itaconate, because what usually is done is that the small molecule is immobilized and the protein is then flown over the molecule. But we know that if we modify itaconate, we will very much modify its properties and change it. And here Bruker has an amazing technology where Johannes Emringer helped us, where you can actually immobilize the protein on the chip with a his tag, and then the protein is in a close proximity to a fluorescent probe. And then you add your analyte, so the small molecule, to the immobilized protein. So we are basically now studying natural itaconate. When the metabolite interacts with the protein changes conformation or moves closer or further from the fluorophore, which changes the intensity of measured fluorescence. And so when Johannes did this analysis, what he saw was that malonate showed absolutely no effect on the peroxiredoxin 5, But in the case of itaconate, we see beautiful decrease in fluorescence in dose-dependent manner, which suggests that itaconate is indeed interacting with peroxiredoxin 5. And the really cool observation was that when you then remove the itaconate from the system, what happens is that the itaconate actually dissipates away from the protein, and this tells us that the interaction is actually non-covalent, meaning that the itaconate is not modifying cysteines of the peroxidoxin 5, it's interacting with it softly. Which was really amazing, and this helped us immensely. And so now I think we can say that indeed itaconate does interact with peroxidoxin 5, it inhibits it, and we believe that it's due to the inhibition of peroxidoxin 5 by itaconate that the boost of interferon happens. But of course, we need to verify this in cells. And so to that, we basically use genetic methods to change the expression of peroxidoxin 5, and we overexpressed it. So you can see it's much higher, the expression level in the overexpressor cells compared to controls. And this alone is enough to prevent the itaconate-mediated boost. So overexpression of PRDX5 blocks the boost of interferon by itaconate. And also what we saw is that PRDX5 overexpressing cells produce less itaconate, less interferon upon DMXAA treatment, which tells us basically that PRDX5 is a regulator of STING activation. And then we also used PRDX5 knockouts. So these are cells that lack PRDX5 in them completely. And we see that indeed, in absence of PRDX5, the interferon production is higher upon DMXA stimulation, confirming with the opposite observation to overexpressor that indeed PRDX5 is a negative regulator of interferon production induced by STING. And it is also true in vivo. So again, we challenged mice with DMXAA and looked at the interferon production upon 6 hours, and we see that PRDX5 knockout has much higher interferon. And for these to kind of fully see if indeed itaconate and PRDX5 are connected, we created double knockouts, meaning mice that like IRG1 and PRDX5. And perform this experiment, and you can see that the wild types in black are dying upon the MXAA challenge. IRG1 knockouts, as we expect, are protected. PRDX5 knockouts may be more sensitive, but we don't see this under these conditions. And when we have IRG1 PRDX5 double knockout, they actually start dying again. So removing PRDX5 on IRG1 knockout background basically reinstates the sensitivity to the DMXAA-induced lethality, which is really proving, I think now, strongly that the itaconate-mediated boost of interferon is actually driven through the inhibition of peroxidoxin 5 and sensitization of STING signaling. And so to kind of conclude this part, what we now know is that we have itaconate, which is a mildly electrophilic molecule. It is an SDH inhibitor, and it's also inhibitor of peroxidoxin 5 that we just found out. Then we used malonate as a control. So this is not electrophilic molecule. It doesn't inhibit peroxidoxin 5, but it does inhibit SDH. And we were thinking, can we find something that is going to basically be non-electrophilic, will not inhibit SDH, and will inhibit peroxidoxin 5 to obtain a molecule, be like an ultimate thing. And so we didn't actually have to go far. All we needed to do was to remove this unsaturated bond in itaconate, and that's 2-methylsuccinic acid. And so this is a non-electrophilic mimetic of itaconate. And when we measured the activity of peroxidoxin 5 in presence of methylsuccinate, we actually see that it is inhibited, just like in the case of itaconate, which was very nice. And then also Johannes was able to measure the interaction of methylsuccinate with PRDX5, and we see a very nice interaction that is indeed, as one would expect, non-covalent. And when we looked into the functional and interferon things, we treated cells with these metabolites, and we see that on the RNA level, interferon-stimulated genes are actually upregulated in presence of both itaconate and methylsuccinate, but not in malonate. Interferon is boosted by methylsuccinate, just like it is by itaconate. And this boost of interferon is indeed driven through ROS, as we see with mitotempo, and also STING, as we see with the STING inhibitor. So methylsuccinate can very nicely phenocopy itaconate, and all it needs is the inhibition of peroxiredoxin 5 to do it, which basically tells us that the electrophilicity and SDH inhibition is not the main driver of the boost. And so, as I mentioned, we had two major phenotypes here. So the question then was, what is the role of potential PRDX inhibition in the inflammasome activation? And for that, basically, we took macrophages, wild type and IRG1 knockouts, and we activated the inflammasome. And so we have standard inflammasome activation, which requires short LPS priming for 3 hours, where we expect no itaconate or very little to be present. And you see that there is no difference between wild type and RG1 knockout. However, in the prolonged treatment activation, where we expect high levels of itaconate, we see that the inflammasome is completely inhibited in wild type, but is running in IRG1 knockout in absence of itaconate. And next, when we reintroduce itaconate into the system, you can see that it basically, again, tolerizes the inflammasome, and it doesn't release IL-1 beta anymore. And so the question was, if we apply methylsuccinate to IRG1 knockout instead of itaconate, can it also rescue the inflammasome inhibition? And indeed, this is what we see dose-dependently. Methylsuccinate was able to inhibit inflammasome activation in IRG1 knockout, telling us that also inflammasome inhibition can be related to the peroxiredoxin inhibition. And this is Gauss-Dermid cleavage, basically showing exactly the same thing. And so to summarize and to conclude, what we now believe is that macrophages are known to produce large quantities of ROS in their responses and in their activation, which leads to cysteine oxidation and other changes. Peroxiredoxins are there to basically decrease the ROS burden in the cell. And we believe that itaconate actually evolved to inhibit peroxiredoxins and preserve and sustain the oxidative environment in the cell, which in turn then translates to its immunoregulatory function and the enhanced interferon and inhibition of inflammasome. And this was, of course, a huge collaborative endeavor. I'm very thankful to Johannes and Brucker for really allowing us to perform the measurements of protein small molecule interactions, which were really amazing, and other people from Artemov Lab. And I thank you for your attention. I'm happy to answer the questions.
Thank you very much for this great talk. Yeah, the floor is open for questions. Please feel free to use any of the chat functions you see, or also use this raise hand function, and I can unmute you. And I will directly start with a question that involves the quote. I will just read out the quote. The question is whether there's any progress with the effect on PRDX3. So the quote is, I have reason to believe, or I have experimental proof that is not yet perfect, that PRDX3 is not affected. And this opens huge avenue of novel research on multiple fronts. It remains to be seen whether it is also an MECFS. So Thomas, could you comment on whether there's any?
We are currently working on this. I have very preliminary data that is suggesting that the effect is weaker, though I can't really confirm it right now 100% that that would be the case, that the PRDX3 is not inhibited.
Okay. And then maybe you can comment on whether there are also other immune cells that produce itaconate in a similar fashion, or is this very specific to macrophages, or can you also see this in like similar effects in other immune cells?
Yes, that's a really great question. So we know that basically itaconate is produced in myeloid cells, meaning that we will see this in neutrophils. We see it in dendritic cells. So indeed, itaconate will be present in all of these cells, and it's actually now very exciting kind of to go in there and see what will happen if the effects are the same. We know that these cells differ in the signaling pathways that they express. We know that, for example, neutrophils like CGAS, though they express STING. So there may be differences in different immune subsets, how they will respond to itaconate and what the mechanism there will be potentially. But we are working on it right now.
Would this also be something that, like in the long term, would have implications for like immunotherapies? Like, can you see any like applications in that direction?
We are currently working on a project that involves cancer. Itaconate has been described to have an effect in cancer. We just are not convinced that the mechanisms so far proposed uh, mm, are the primary driver, to put it that way. So I think there will be kind of like overarching things going to different diseases. We obviously would kind of expect itaconate to have effect in viral diseases, which we do see, and potentially, prophylaxis with methotrexate could be beneficial there. And in cancer, we do see that itaconate is detrimental. Actually, it's not beneficial to have it, and it seems to inhibit immunotherapy. So then there is a potential for inhibitors to be involved for itaconate production.
Mm-hmm. That sounds cool. I also know that, for example, for T cells, also the whole like ROS activation pathways are very crucial also for their fitness, like whether it's endogenous T cells or also if we think about like TCR therapies, CAR T cell therapies. So yeah, there's for sure a lot to discover still.
T cells are not known to produce itaconate, so under normal circumstances. So there's a lot of research now being done into itaconate uptake, which is very interesting. It's very intriguing, saying that like macrophages or other immune cells would release itaconate into the stroma, and that will be getting uptaken by other cells. There are challenges with the fact that itaconate, as we believe, requires quite higher concentrations, which you don't normally reach within the tissue. But yes, this is actually very exciting, like what other effects it has on other immune cells outside of just myeloid cells.
Mm-hmm. So what would be like your priority next steps for this project? We've already talked about like PRDX3, but also other immune cells. Like, what would you say is what
we are basically looking to like already peroxiredoxins. That's the next step. Next big step that we are working on is to essentially looking into other phenotypes that we have for itaconate, if they can be mediated through the inhibition of peroxiredoxin 5. And there are a lot of things we don't know still. Like we don't exactly know how hydrogen peroxide boosts STING. This is not something that is known, and we haven't actually addressed it in the paper. So this is something we are also working on to really pinpoint the mechanism how this happens. Just by the ROS basically boosting interferon even outside of itaconate. So there are a lot of questions that we have at the moment.
Mm-hmm. There's another question about the benefits at the cell level. So what is the response, variable growth or any metabolite, and whether you have collected RNA sequencing or proteomics data to evaluate whether the cysteine oxidation affects other parts of the metabolism?
Okay, so if I understand it correctly, is like what are the other changes induced by itaconate? Am I
I'm also not
getting it?
very sure about the phrasing. Maybe the person asking the question can help us out. Ah, yeah. Wait,
I
I will
can.
unmute you.
Oh yeah.
All right. Hi. You should be able to unmute yourself now and
Oh,
talk.
perfect. Thank you.
Hello.
Can you hear me? Can you hear me now?
Yes.
Oh, perfect. Yeah, no, I was wondering, when you go to in vivo or to mice, you can clearly see the benefits, right? But
Mhm.
at the cell level, what is your response variable to say, oh, this is working or not? Like, do you actually cells not losing number of cells, or like a measuring uptake rates of glucose, or what does it tell you that the cells are doing okay or not?
I mean, we measured survival. We measured basically levels of ATP. We measured. We used two different methods to measure cell survival. So cells are fine. We know that they're okay up to 7.5 millimolar itaconic acid. Uh, then there is some level of toxicity induced by itaconate after when you go higher than 7.5. So that's why we are staying in the 5 millimolar range, which is safe even for 24 hours. Macrophages are fine. We don't observe any signs of toxicity in them.
I see. Good. And then, have you collected any RNA-seq or proteomics when you treat with cis to see if there is an effect of all these 6-day effect, not only
We
in
yeah,
PRT?
we run a number of proteomic analysis. So one that we did is the itaconation, right? So where we look at all the proteins, and as it is with when you are basically looking only at a natural itaconate, either endogenous or exogenous, we see roughly 30 proteins that are modified by itaconate, which is completely different story when you look, and people usually use dimethyl itaconate or 4-octyl itaconate to look for the itaconated proteins. But the problem is that when you increase electrophilicity of the molecule, you are naturally going to increase the number of modified cysteines. But it doesn't tell you if it is a real modification you're also going to see with natural itaconate or not. So we really see small number of proteins, and we see them very robustly in like number of analyses that we did or that other groups did. You can see these proteins coming up. So there is a change. However, I didn't show the data here, but what is a really huge change proteome wide is the oxidation of cysteines that is due to increased oxidative environment. So there we see really a giant proteome-wide effect of itaconate that is manifested, and I think that's more important than the actual itaconation itself.
Oh,
This
yeah,
effect of
because
itaconate.
yeah, that may be due to the you're poking the TCA cycle, and that is the source of a lot of other pathways, right?
Mm-hmm.
So it may be because of that. Oh no, this is very nice. Yeah. And my last question, well, now that I have them, is that so we have work on reverse vaccines, and these are. Do you see itaconate become part of one of these reverse vaccines and potential treatments?
Vaccines.
I don't know if you're familiar with this concept of reverse vaccines. and they are polymers of succinate that
Mm-hmm.
we are using to treat rheumatitis and to see the effects on this. Yeah. So itaconate is a very similar structure, right? And then you, so
Mm-hmm.
right now, are
Yeah.
you providing it as a polymer or are you
No,
providing it as a mono?
no. We are actually just putting natural itaconate solution for the in vitro treatments. It is natural itaconate solution. If we administer itaconate in vivo, it's just neutralized itaconate, but we don't polymerize it. No, but it's a
Oh,
very interesting idea. Just kind of see what would happen with a polymerized itaconate. I like the idea.
yeah, I might reach out to you. Thank you.
Sure.
Bueno, you got your my
Yeah, please
WhatsApp.
do. That'd be great.
Thank you.
Thanks a lot. I think we are also already running out of time. Maybe you can just put up your slide again with the contact details, so in case people want to reach out
Yeah.
about
That,
anything.
I have to share.
And if we didn't get to your questions, we'll make sure to reach out afterwards also. Feel free to also contact us at Broker Biosensors. If you also want to try out the system, we actually also organize technology demonstrations, so you can just try out the technology with your projects, with your molecules, see whether it works and whether it brings any value to your research project. We are always happy to do these demonstrations. And with that, I think thank you again very much, Thomas, for your talk. Thank you all for participating for the discussion. I hope to see some of you in our upcoming episodes of this webinar series. And you can also check out recordings of previous episodes on our YouTube channel. And with that, I wish you all a pleasant rest of the day. Bye bye.
Bye.